Right. So Sumir Panji obtained his PhD in bioinformatics from University of Western Cape as part of the Stanford South Africa Biomedical Informatics program, where he developed computational and analysis pipelines to determine the intersection between bacterial virulence and position selection in Professor Winston Hyde's laboratory. He completed his postdoctoral studies in Professor Alan Christopher's laboratory in South Africa National Bioinformatics Institute, SANBI, where his focus was on genome assembly, annotation, determining large-scale statistical analysis of genomics data, and development of various computational pipelines and analysis workflows for various genomics data types. Dr. Panji's main interest is in creating and implementing computational analysis workflows, statistical analysis of biomedical data, biological algorithms, high-performance computing, and overall application of bioinformatics in genomic methods to better understand complex biological systems. Dr. Panji is currently a bioinformaticist within the H3 BioNet Consortium, who is interested in genome science, data analysis workflows, statistical analysis of large omics data, implementation and interpretation of bioinformatics solutions to diverse biological problems, and providing bioinformatics support to H3 Africa projects. And is passionate about developing bioinformatics capacity on the African continent. And I think that's what he is here with us today to do, to develop capacity in bioinformatics for all of you curious minds, ready to learn, willing to learn. So thank you so much and welcome to me.
Thank you, Caleb. Thank you for that introduction. I hope my volume is clear.
Yes, we can hear you well.
Okay, great. So I'm just going to start sharing my slides. So thank you so much. As Caleb says, I'm a bioinformatist that's based with the H3 BioNet project, and I'm based in Professor Nikki Mulder's lab, and currently working to provide support for the H3 BioNet, H3 Africa projects as part of the H3 BioNet grant. So today we're going to go over Unix, Linux. I'm just going to share my screen. There we go. So I'm just going to, today's, it's just going to be a lecture, and I'm just going to go over a lot of commands over Unix and Linux. And first off, I'd just like to say thank you to Dr. Amal Gulia, who's helped develop a lot of these core materials as well with me. And we've adapted these materials from the Introduction to Bioinformatics Training, NGS course, which is the H3 BioNet IBT course, and the Welcome Trust H3 BioNet NGS Bioinformatics course, Africa 2021. I see some people have their hands raised. Is there a question?
I think those are legacy and
Oh,
you
okay,
just
cool.
lower them, yes.
Right, I was thinking it's just the intro slide. Okay, so today we're going to go, I'll just give you an introduction over Linux and how you usually interact with Linux, your types of input and output. I think you'll cover the input output a lot better when you do the HPC stuff, and it'll make more sense then. And then just your basic command line structure for Linux commands. When you're working with the Linux system, I mean, navigating through the file system is very important, especially finding where files are, because a lot of your files will be the input to your bioinformatics commands and applications that you'll be doing. So navigating through a file system is quite important. And then I'll provide some commands and examples with that as well. And then within Linux, you'll usually do work with files. So I'll provide some commands with files in there and how to edit files and permissions. And also you want to see what's inside the files as well when you want to process them. So looking at file contents and examples of that as well and some commands there. A little bit of searching for commands and searching for patterns and examples. We'll do a little bit of grep, and grep is really useful, but I think we'll do a lot more of the pattern matching when we do the sed and awk part as well tomorrow. And then some Linux commands for extracting context from a file and for examples, and then output redirection of Linux commands. And then how do you combine multiple Linux commands, because that makes your work a lot more efficiency, and some useful tips as well. And then there's a practical session which I'll upload today, and it's just to go through some of these commands, which I'm going to cover right now. So let's go through some definitions first. So you have an operating system. So an operating system is just basically a system that comprises of software, and it tends to manage your resources, such as your hardware and your applications in there, like your programs. So a lot of you will be used to your Windows operating system. So the base OS is Windows, and then you have your applications like MS Word and MS Excel and stuff like that. You have different operating systems such as Unix and Linux as well, which do have their own applications and stuff. And then you within the OS, you have what's called a kernel. A kernel is just a computer program that's the main layer between the operating system and the hardware, and it controls a lot of the system's functions. Usually when people interact with Unix and Linux, they use what's called a shell, and a shell sometimes is referred to as a kernel, although it's not technically a kernel. So Unix and Linux, what is it? It's just an operating system. So Unix was originally developed at Bell Labs for AT&T. It's closed source, and it's usually paid for software. Sorry, yeah. Okay, let me switch off my video because I do have bad lighting. So let me switch off my video. There we go. And let me just move this chat quickly because it's in my slides. Okay, so it's a computer operating system that they use for a lot of processing of text and stuff, and it grew in popularity, especially when the universities tend to take it on. And you have variants of Unix that include Linux, and such as a Mac. If you're using a Mac PC, then you tend to have a Mac OS. And Linux is an open source type of Unix operating systems. It's based on the Linux kernel that's created by Linus Torvalds. And you have examples such as Ubuntu, Fedora, Red Hat. And the reason we use Linux is that a lot of our biological data is in text format, or we have tools that access them, such as your FASTA files, NGS data, such as the VCF files Sean was showing you earlier. You have other file formats, such as your BAM, which is a binary alignment format, and that's a binary file, and so only the computer can understand it. It's not human readable. But also your results, when you process your data, the output you get will be in text format as well. So how do you manipulate that? And if you're dealing with like maybe 10 or 20 or 50 sequences, it's easy to do it by hand. But a typical NGS human genome will probably have over 3 million reads, well, more than 3 million reads. So, I mean, how are you going to work with that? And of course, the bigger the data set you have, the more statistical power you have as well. So Unix and Linux are just very efficient when it comes to working on big data sets, and especially with numerous files and having to move it out to other applications as well. It has a powerful and flexible suite of commands that can be used to process and analyze data as well. And especially if it's text-based data as well, it's a lot more quicker than trying to open a file and read it in a Windows application. Nice things about Linux commands is you can actually combine them. So you can change the output of one command is the input to another command using pipes. And if you look at most HPCs, it's actually Linux and Unix is the standard operating system on most clusters, such as the one you log in today as well. It's similar as the way Microsoft Windows is the dominating operating system on PCs as well, and also in the commercial sector. Because Unix Linux is used by a lot of academic institutions in high performance computing, it's really optimized to be above really good best multi-use and multitasking OS, especially like on clusters, which is when you log in today. And it works really well with scheduling systems as well. And it's wherever you go, it's probably the most preferred operating system for large scale computing, especially in the scientific domain. So if you're going to access Unix Linux, the main way you're going to do that is using a terminal. So terminal is a wrapper program which runs a shell. Most of the shells you have are known as born again shells or bash shells. So when you log in today to the HPC, first thing you'll use is probably SSH, and then you'll open a shell and then interact with that. So you have the shell prompt, and on that you have your username, so it'll tell you who you are, and your user account. So mine is user 0, whereas yours will be user 1 to 26. You'll have your username and you'll have the machine name as well. And most of the interaction you'll do with Unix Linux is actually typing on the shell. So do get familiar with typing some of the commands. You don't have to be an expert on them, but the more you use it, the more familiar you get with it. It's like learning a new language as well. So when it comes to interacting with Unix Linux, you have 3 types of output. You have your standard input. This is where you input your commands, and this could be from a system. And most of the times we do this using a keyboard, and that's known as your standard in. The standard output or standard out is where the results of the input commands you're given go to. So for example, when we say standard out, that usually refers to the screen. So if you do like a cat or a lesson or file, to look at the contents, that file will be printed onto your screen itself. Or you can redirect that output of that file into a new file if you give it a redirection command, such as the one shown below here with the greater than sign. So your standard error is any error message that occurs from executing the command. So if you do like an ls and you don't give it a file name, or if you do like a less and without a file name, then you'll get a bit of a command hanging there and you'll get an error. So I think the standard error, you probably cover that a lot more when you do the HPC stuff, and this will make more sense then. So just do bear this in mind. So with Linux, it has a specific command line structure. You have your command name, then you have what's called a minus or switch, and then your options, and then your arguments. And then your arguments are usually your input file or what you want to do. For example, this example here shows grep -c input fastSave file. This command, if you try this, is going to hang because there's an error here, because I've not given it a pattern so far, but you'll see that in the further slides for grep. In order to learn what options and switches you have for command, Unix has what's called a manual. So you type man and then the name of the command, and that will give you the options that you can use with grep. And sometimes they usually also have examples as well. Sometimes if you enter a Linux command without any options or need to exit the command, then a quick tip is just to, because what happens is then the program tends to hang looking for input or waiting for your user input. So you can just type Control-C. And one good tip about learning Linux is there's a lot out there and a lot of different ways of doing the same thing. So it's really good to go out and look at example boards like Stackflow and message boards as well. And you get a better idea of how people are using the commands and what they're using it for as well. And that's the best way of actually increasing your knowledge, because it's like learning starter code and looking at different examples, which then you can try out yourself. So when it comes to Unix, what does one deal with? Usually you deal with directories and files. So the basic file system is you have a root directory of the entire file system, which is your root there. And then you have a user's home directory. So your home directory is what you'll probably be logging into today on the HPC. And then you have other types of directories, such as device files, such as your binaries. The binaries is usually where you store all your executables, all the programs that are available for the system users as well. And then you have your configuration files as well. So within the user's home directory, you would have user 1, user 2, user 3, and you probably have a look at that today when you log in. And then within each user account, you can have multiple directories, such as your desktop, your documents, Linux. And within each directory, you can have multiple subdirectories as well. So on the top level, it's known as a directory. As you go down, it's known as subdirectories. And then you also have files within those directories as well. So this is similar to like your Windows system, where you're going to click on a window and navigate with the GUI, but just doing this, you're going to do it on the command line. So as I said, within each subdirectory, you can have different files as well, which are your ordinary files, and you have directory files. So navigating through these directories is a bit tricky. So when you log into the Unix system, the working directory is your home directory. So when you do that today, just type pwd and you'll get an idea of where you are. And while you're working on a Linux system, you'll be associated to one directory called the working directory or your current directory. So pwd means print working directory, and that's your current directory. And the reason it's important to remember about directories and navigation is because usually you're going to be working with files. Your input files will reside in one directory. Your executable will reside in another directory. So you'd want to point your program to where the files are as input files. And of course, once you point a computer program to your input files, it's going to generate output. So you want to also push that output to somewhere else where you can then work on that output and analyze it. And when you type pwd, an abbreviation of the working directory is displayed as part of your prompt on your terminal, and that you'll see that today as well. And you'll get your absolute path of your working directory if you type pwd, such as home user2 genomics. So when you talk about paths, what's a path? A path is locates a given file in a system hierarchy. An absolute path for file system hierarchy is given in a folder and describes the paths all the way to the root. So for example, I have slash here. So each slash means you're going up a directory. So if I do slash home/user/genomics pfile_syscrum. That will take me to where this file is. In order to do that, you can put a CP there, you can do a CD. A CD means change directory, and a CP means copy a file. But we'll go through some of those commands in more detail. And a relative path just describes the path of the file starting from the current working directory. So if you're in that directory genomics already, then you'll just be saying genomics pfile_syscrum. So it'd be like ls on that. And if just to illustrate this a little bit more, so here's the top directory, here's your home directory, your user2, your course data, your Unix, your practical, and then your Unix PDF. So the path to this Unix PDF, which is the full path, is /home. And every time you use a slash, it means go up one directory, and then /user2, and then /course data, and then practical, and then Unix. So that's the full path to there. So there's some useful commands. So I did mention CD. CD means change directory. It allows you to move from one directory to another. And that's kind of useful if you're looking for files or if you're using an executable and you want to point it to where your input files are. And then the next command, which is quite useful, is also ls. But just to go back with the CD. So if I do CD .. that means change to one directory up. If I do CD .. CD .. means change to 3 directories up. And CD tilde means move to your home directory as well. And yeah, the CD command is really useful when you're looking for files and where you want to find the executables and where you want to point stuff, especially if you want to copy stuff as well. The next command that's quite useful is once you're in a directory, you want to see what files are in there. So you use the list command. The list command is known as ls. So it lists the contents of a directory. So some useful options is if you use ls-l, it shows you the modified date and time, or the file and folder name, the owner of the file permissions. I like ls-lh because that shows you the file size in human readable format. ls-r, it recursively lists subdirectories. And one command which I'd really like using is like, similar to the CD .. I use ls to do that when I'm looking for files or if I want to get the path of a directory. So when you do login, try doing the ls // just to see what other directories are in the system. Sometimes you want to make directories because you want to put your input files in there. So make mkdir means make directory, and it's just simply type in the name, the command name, and then the directory you want to make. So for example, data files, and it will make the directory for you. You can give it a path, or usually when I make a directory, I do that when I'm working in my current directory. But you can also make directories in using the other path structures as well, which can be efficient as well. So as I said, the new directory will be created in your current working directory if you've not specified a path to where the directory should be created, such as make directory and then directory name and then the path of where you want that to be. You can remove files and you can remove directories. So rmdir removes a directory if it's empty. If not, you might have to go into the directory itself and just do an rm-r to remove the files, and then you do an rm-dir. Or you can do an rm-dir-f, which means to force it. So the command structure will be your command name, and then your directory name, and then your path. If you do that, the directory should be in your working directory, in your current working directory, and that you get by typing pwd. If you want to move from elsewhere, you have to specify the path. Similar like when you make the directory, you have to also give the path of where you want to move it. And as I said, if the directory is not empty, you'll get an error message, which directory is not empty. I think you have to be a bit careful with the rmdir command, because once you delete files in Unix, then they are kind of permanently deleted. The only way you can recover those files is if your system administrator keeps a backup. So be very, very careful with the rm command. So you can copy, move, and remove files. So CP is a copy files from and directories. So usual syntax is your CP path from and path to. So example here is CP, and then the /home user2 practical Unix PDF is the path there. And then I put a dot over here at the end. This dot signifies copy the file from this location into my current working directory. And it's usually good practice to copy files into your current working directory if you are working with them, or if you, as you get more familiar with Unix, then you can copy them into directories which you create without having to be in there. Another way of doing it is you can use this syntax where you cp from home. So I'm copying the file Unix PDF from my home directory user2 under the practical. I'm copying it to the home directory for user3 practical. You can use the move command to move or rename files and directories. So that way you don't keep copies of the file. You can say move home user2 practical Unix PDF to your next username over there. And this is just an example of the remove files and directories. So rm home user2 practical Unix PDF. So that's basically how you can copy, move, and remove files within the Linux system. Maybe we should take a break here. Are there any questions at this point? Okay, I don't see any questions in the chat, so I guess I'll continue. So the simplest form when using the copy command is cp file1 to file2 to copy the contents of file1 into file2. If file2 doesn't exist, then it's created when you invoke this copy command. Otherwise, if it does exist, then the problem is you'll actually overwrite the data that's existing in file2. So maybe sometimes you want to use cp minus I, which is copy and interactive, so you don't write the file. You can copy the file name and you can give it a directory path, as you'll see in some of the practicals later on today. And you can copy the file or directory to a specified destination directory. If you're going to copy a directory, then you'll use the cp minus R for recursive, because then that way you actually copy all the files within that directory to a new directory. So as I said, to prevent overwriting the files, one can use the interactive mode with the cp command and use the minus option I. So for example, if I use cp minus I, file1 faster file2, but if file2 exists, the user is notified. So you get a warning. Do you really want to overwrite the contents of your file and lose the data there with the contents of the new file or not? So it's a useful switch to use when you've got files that the name more or less the same way. And then you can do cp minus R, so the contents of the directory in directory 1. And if directory 2 does not exist, it is created. Otherwise, you create a directory name directory 1 within directory 2. And this will be a bit more clearer when you actually do the practical. But if you do want to copy multiple files from a directory instead of one file, then you'd use the minus R switch for recursive. So the move command moves or renames files and directories depending on how it's used. So to rename a file, I'd use move file name 1 to file name 2. So I see a question from Winnie Mumbi. Why use a Unix shell instead of a Windows shell? So I think with Windows, when you use a Windows shell, are you going to be using a Windows shell that's using more or less a Unix variant? Because I know with Azure and stuff, you can do that. We do have more of a Bash variant. But traditionally, Windows has tend to be more like on a DOS operating system. So the commands you tend to use on Windows are very different than what you use on Unix, especially the syntax and stuff as well. And I think a lot of the scientific programs and applications that you get for bioinformatics and also scientific computing tend to work on Unix operating systems as well. And probably because Unix operating systems tend to have really good schedulers and stuff, especially when you're doing high performance computing. And especially when you have lots of people that are using the same compute resource as you guys would probably be using today. It's just much easier to manage accounts and user creation with Unix and Linux than it is with Windows. Hopefully, Winnie, I've answered your question. Great. So to rename a file, you'd say move file name to file name 2. If file 2 exists, its contents are replaced with the contents of file 1. And move is different from copy, because with copy you're creating a duplicate of that file. With move, you're actually moving that file, so you won't have a duplicate of that file. So you can also use the interactive mode. So move -i, so which is a -i switch, file name 1 to file name 2. If you want to move a file to a directory to another directory, you do move file and then you give it the directory path. Or to move different files to a directory or another directory, you can do move and then you can give it all 3 file names and then the directory path to where you want to move it. And similar with the directory, you can move the whole directory by saying directory 1 to directory 2. If directory 2 does not exist, then directory 1 is renamed to directory 2. If directory 2 does exist, then the directory 1 is moved to within directory 2. So this is a bit safer than CP. But sometimes you'd want to use the move command, because sometimes if you're working with large data sets, you don't have multiple copies of the large data set, because it does take a lot of computational storage and memory as well. So sometimes you rather move files than copy them and keep copies there. When you're working files, I mean, sometimes you do get a lot of intermediate files and some files that you can use to delete. And in order to delete a file, you'd use the rm or remove command. To remove many files, you can give it rm and then a list of file names. So that can be file name 1, file name 2, all the way to like file name 100 if you like. Another good way of using rm is actually to use the interactive mode. So use the -i switch. This then will prompt you to ask you, do you really want to delete these files? And then you can go ahead and do that if you're sure. And then with the delete contents and directories, you use the rm-r recursive command, and you go through the directories itself. And as I said before, just be a bit careful with the rm command, because Linux, unlike Windows, does not have a recycle bin or an undelete command. So if you delete that data, it's kind of gone for good, unless your system administrator keeps backups. And then you'll have to go to them and ask them to restore your system from that last time it was backed up. And this does happen to all of us sometimes when you do rm and then star, and it just deletes every single file in your home directory. So just be a bit careful with that, especially the wildcards, because you can really inflict a lot of damage there. And before here's a good trick to try. Before using the rm command, maybe construct your command using ls, so you can actually see what the files are themselves. And sometimes it might just be useful to actually copy those files into a directory itself and then go into the directory and delete all those files, just so you don't have any unintended consequences. If you use a wildcard and by mistake, it pattern matches something else which you're not sure about. So in Linux, there's really no distinction between the names of ordinary files and the names of directory files. You can have multiple files that have the same name, but not in the same directory. So if you're working in a directory, you can have fasta1, and the directory above you can have fasta1, but both of them will not be able to operate in the same directory. So you have to have unique names. A good tip that we tend to do in Unix Linux is tend to have underscores and don't leave spaces between file names. And the reason we do this is because when you're typing out commands and file names, without having spaces, if you hit the tab key, it auto completes the file name for you. So that really saves you a lot of time. And Linux is case sensitive. So coarse data and coarse capital data and capital coarse data are very different and would represent 3 distinct files as well. And so that's why it's useful to actually use the tab completion and keep underscores between your names, just because in that way you don't have spaces. In most cases, file extensions are optional. So you have a .txt, a .exe if it's executable, a .py if it's a Python script, and lots of other types of extensions like a .foste. And as I said, if you really want to work quickly in Unix Linux, hit the tab key after typing the first few letters of a file name, because that will help you. Well, it'll complete the file name for you and save you time in typing. As you'll see with today's practical, because the file name we're using called plasma.db is quite big. So when you're working files, you have what's called permissions. So your permissions, you can view your permissions if you type ls-l. And if you look on the left-hand side, you have what you know as your permissions. So that D means it's a directory, and that one here without a D means it's a file. And then you have your file permissions and your user permissions, which is your read, write, and executable for the user. And then you have the group permissions, which is read and execute for everybody else, and read and execute for everybody else. Usually you get your file sizes in bytes. You get to know which group you are in. So you'll be in the user group, I'm guessing today. And then you also know who the owner of the file is as well. When you do this, because you're doing it in your home directory and the file is in your home directory, you'll be the owner of the file. And then you also have the file names, which is given over here. And then you have the time that the file was modified as well. So the ls-l is a very useful command, which we tend to use quite a lot. Your permissions are broken into 4 sections. You have your feature type. So a slash here indicates it's a file, similar to here. D indicates a directory, and then you have something called an L, which is a symbolic link. Sometimes we link, we create paths to other programs or other data stores, which are known as symbolic links as well. It's not the full path there. And that's usually when you're having stuff that's used on a system-wide level as well. You have your user owner permissions. So your R means your read, your W means your write, and your X means your execute. And execute is quite important because if it's a program, then you want it to have executable permissions, especially if it's like a Python script or something. And so sometimes when you download applications and install them, you need to actually turn the programs into executables as well, which you can do using the chmod command. You have your group permissions. So in this case, the group is able to read. They're not able to write and execute permissions because they don't have the W or the X. And then the read, write, and execute permissions for other users on the systems as well. So this is your user, this is the group one, and these are other permissions as well. And we'll use a bit of the chmod command, so you can have a look at this in practice when you do the practical today. So the chmod command is used to change the permissions of a file of a directory. So your syntax is basically change modification, and that's known as chmod. Then you have your options, and then your permissions, and your file name, your input file name. And only the owner of the file can use chmod to change the permissions of that file if it's in a directory. And permissions define the permissions for the owner, the group, and the users, and anyone else. And 2 ways to specify the permissions. You can use the symbols, or you can use octals or digits. I'm more familiar with the digits, so I tend to use chmod 755. So if I use 755, that would give me this kind of syntax, where I'll have read, write, executable permissions. The group will have read and executable, and others will have read and executable permissions as well. The main way you're going to interact with files, if you want to edit files, is using a text editor. So I'm not going to go too much into text editors today. You might probably use them when you're creating some of your qsub commands on the HPC. But a text editor that's on most Unix systems is known as Nano, which is a simple and easy to use text editor. So you just type Nano and then the name of the file, and then it'll open the contents of that file in the text editor itself, and then you can edit there. When you're working in a text editor, it's mostly command line. You can use your mouse, but the GUI interfaces are not that great. So you do need to get a bit familiar with a lot of the keyboard syntax and some of the commands. If you see, when you open a shell, you'll see something like a top hat. So that top hat means control. So if you want to exit a file, you type Control X, and then it'll ask you to save it, and then you type Y or N for yes or no. The other editors, such as gedit, which is also quite easy to use. And then you have more suitable text editors, which if you're writing computer programs and stuff, people tend to use a lot more, such as your Vim or your Emacs and Genie. But they do require some learning, and it's really good to learn them when you're learning how to program in Python or something. I think Python also has its own editor. So when you're working files, you like to view the contents of the file. So I'm just going to give you some examples of commands that we can use to view the contents of the file. So if it's a small file, you type cat and then the file name. So cat means, well, just print out the file contents itself. And then the contents of the files will be printed out to your screen. If it's a small file, it's fine because then it usually stops printing quickly. But if it's a file that's got like over 2 million lines, you'll probably be sitting there for quite some time while it prints out each since every line in that 2 million line file. More, you can use more to view the content of a long file and navigate through it. So you just type more and then the file name and foster. The command I tend to use a lot more when I'm looking at file contents is less. And also because less has some really nice options where you can actually look at NGS data as well. And the nice thing about less is when you view the content of a file, you can do it by portions, and then you can also navigate through that file as well. I'll give you some examples of that. And then you have the head command where you can view the first lines of a long file. And that's usually useful if you don't look at all the file contents, but you just want to have a quick peek to see what does it look like. For example, you sent your sequences into a program and you've got some results out and you want to see, do the results seem okay? Rather than looking at the whole file, you can just have a quick peek to see on the top of the file. So you type head and the file name. If you don't give it an option, it usually prints the first 10 lines. And if you want to look at the end of the file as well, just to make sure that all your sequences are in there, you can use tail as look at the last portion of the file. So I prefer the less command because it displays the text file content one page at a time, and it kind of differs from more because it allows you to scroll backward and forward as well. There are some arguments why less is more efficient than more. Is because sometimes more has to read the whole file in memory, and if it's a big file, it can take some time, whereas less doesn't tend to read the whole file in memory. So sometimes it tends to be a bit more efficient. So I would say if you learn less, do learn it, and it's a really useful command. So you can move a page down, either use the page down or the down arrow or the up arrow on your keyboard to go through that. You can also use the page up key and the page down key as well. If you want to exit viewing the contents of a file when you're on less, just type Q and that will get you out of there. If you want to go right till the end of the file when you're viewing it in less, just type G. And the nice thing about less as well is you can also find a pattern in a file. So when you type in less on your file, you just hit the / and then the pattern name. So for example, /ATCG or /NAME, and then that will highlight that for you in the file as well. So the head command kind of displays the text file content by content, and by default, it's the first 10 lines that will display. You can change that behavior by using the -n option, which stands for number of lines. So you can give it like head -n 100, and that will give you the first 100 lines of the file. Tail works the similar way as head, but it shows you at the end of the file. And by default, it also shows you the last 10 lines at the time. And as I said, you can use the -n switch to change the number of lines you want to display on the file. So sometimes working with files, you do want to get some kind of statistics. So like, let's say if I've got a file and I know it's meant to have like, 3 million lines, how will I be able to determine that just to make sure it's fine? So you can use what's called the word count command, or WC. And if you use WC -L, will give you the number of lines in your file called file name foster. And that's really useful for checking your data to make sure everything is complete and you have everything in there before you send it for another program to analyze the data. So you have some useful commands, and with all of these commands, I mean, do type man and the command name on the Linux terminal, and that should give you a lot more information about them. And also do look on forums like Stack Exchange as well to see how people use this. I tend to use WC -L quite a bit. That's just because then I know what kind of counts I'm getting. But you can also use other counting methods as well, which I'll show you. So one of the most popular programs in Unix is grep. It's your global regular expression profile. And this is used to search for the occurrence of a pattern or regular expression in a file. And by using grep, you can actually find a pattern and then you can use it to provide to other commands in Unix to get other information as well. So for example, if I want to extract the lines containing the term sequence from a file, I'll just type grep, the sequence, and then the file name foster. If you want to extract lines that do not contain a certain pattern, i.e. the inverse, then you use the -v option as well. And that will print out every line that does not have the name sequence on there. And when you use grep, by default, the output will go to your screen as well. And that's useful because you can also use that to make sure that it's actually doing, the command's doing what you want it to do before you redirect it to another file. So if you want to search for the occurrence of a specific pattern or regular expression using wildcards in a file, you'd use grep, your pattern, and then your file name. So for example, grep and then I'm using the fastA or the greater than sign, and that will give me all the lines that have this pattern in the file name called fastA. If I want to find out how many sequences are in my file name fastA, I'd use the same pattern to search for that, because by default, each sequence accession starts, or fastA header starts with a greater than sign. So the number of greater than signs in my file would equal to the number of sequences. So I can do grep and then search for the greater than sign, and then -C, and then file name fastA. You can also use the output of other commands into grep. So for example, if I want to find every file, or print the file names that end with .fastA in my directory, I'll do an ls-l to list all my files. And then the output of that I'll send to grep by using what's called a pipe operator over here. And then when it's sent to grep, I'll ask grep to actually look for everything that ends with a .fastA and provide that to me. So these commands will just print out the lines that match the greater than character, which is your fastA headings. And the second one will provide the count of the number of fastA headings within a file. And the third one will print out the file names from the ls command and will pattern match the ones that end with the .fastA. So some other commands which are really useful is the sort command, which reorders the contents of a file alphabetically. You'd use your sort, then your options. Your options would be like your minus switches, so you can have -C or -U. And then you have your file name in fastA. You have the unique command, which removes duplicated lines. So your syntax will be unique, then your options, and then your file name. And all these commands, you just type them out the way they are over here on the screen. And that's pretty nice about Unix, because just learning and typing a small set of commands actually allows you to do a lot of operations very quickly once you get familiar with it. You have join, which is compares the contents of two files and outputs the common entries. And diff to compare the contents of two files and outputs the difference between them, which is really useful because sometimes you might have modified a file and you made a copy of it, and you want to know what the difference is between both of them. And the syntax for that is just your diff and then your options, and then you give it the two file names that you want to get the differences between. So with the sort commands, sort outputs the sorted order for file content based on a specific sort key. By default, it takes the entire input. So if I do sort options file name fastA, it will sort what's in there according to alphabetically. If I give it a -n, it means sort by numerically. And usually sorted files are quite useful because you use them as input for several other commands. So you also use sort with a combination of other commands, such as unique ones. So for example, I'd say sort gene list file text if I had a gene list of accessions, and they might have some duplicate gene accessions because maybe it's some expression studies, or I've got them from different places and I'm combining them to create a gene list that I want to be unique. So I'd say sort gene list file text and then unique to get the unique genes in that file. And remember, Unix is case sensitive. So if my one gene starts with a capital A and my other one starts with a small a, they wouldn't be considered as, they'll be considered as two unique entries rather than actually being the same entry or duplicate. So you do need to process your data to make sure that there is some uniformity when you use some of these commands. And as suggested, type man sort and also look at forums for ideas on usage on this. So unique is also a very useful command that I tend to use a lot in Unix. It outputs a file with no duplicated lines, for example, a single entry per line. But in order to use unique, actually, you need to have a sorted file as input. So you'd have unique options and then your sorted file fastA name. A useful option I do, because usually when I'm using the unique, I kind of want to get counts of what my genes numbers are, what the representation is in a list. So a useful option is to use the -C, and that will output each line with the number of times it repeats within a file. So an example usage is sort your gene list file text, and then you pipe it to unique -C, and you get a count of the number of times the gene name appears in the file. Use the man option as well to have a look at some examples on this. The diff command is quite useful because you can compare two files and it displays the different entries. And you can use it to highlight differences between two versions of the same file. Sometimes you do modify a file and you want to make sure that your modifications are there. So your default output is common lines are not shown, only different lines are indicated and shows what's been added in terms of A, deleted, or changed, C. And its usage is quite straightforward. It's just the command name, which is diff, your options, and then your two file names as well. So when it comes to redirection, using all these commands will actually output your results onto your standard output, which is your screen. So you can redirect the output of the commands to a new file or append to an existing one. And it's useful if you want to pull out exception headers or parts of a file used in other parts of application analysis. So your syntax will be your command options, your file name, and then your input file, and then you direct that using the greater than sign and give it a new file name that you want it to be put in, such as file name .fasta.out. The second file will be created if it does not exist. If it does exist, it will be overwritten by the new output. So for example, over here, grep and this file name fasta will give me my exceptions in a text. If I do that again with fasta file name fasta2 and pull out the exceptions, it will overwrite the existing file. But over here, I've got one with an S and one without an S, so it wouldn't overwrite that file. If I wanted to append to that file, I'd use the double greater than sign. So that means add to the bottom of the file. So sometimes if you're going to work with extract data from multiple files to put in one file, then you'll be looking at the append kind of syntax where you use the double greater than sign. Unix is useful because you can combine multiple commands into a chain of commands. And so you can do a lot of powerful operations if you get familiar with the language itself. And you'd want to perform multiple operations to get to a certain point. So, I mean, for now, because you're starting out, we will run each command individually. But sometimes you'd want to run a series of 4 or 5 commands, and it's quite inefficient and painful to run an ls and then get the output and then run something else on there and then get the output and then run something else. So the nice thing about Unix is you can actually combine the output of one command and redirect it as input for another command using the pipe character, which we covered a little bit earlier. So for example, over here I'd say grep and then grep the fasta headers, sort them, I pipe that output into sort, and then from the sort output I'll pipe that into unique to get the unique fasta headers in a file, or the unique sequences or accession numbers in a file. And I could also even count the numbers and send it to an output file over here. And it'll be stored in a file called output file.txt. So we're coming to the end of the talk. Just some useful tips. Use tab completion. That's your tab key. It will save you time and prevent errors. And I think for today, when you do copy the Plasma DB file, you probably find it a lot useful using the tab there. As a first-time user, do build your commands slowly because it's more about the logic of what you're trying to do. And once you have an idea of what you're trying to do, then just finding the bits of the language that actually fit in and do what you want it to do is a lot easier, especially if you go to like like Stack Exchange and look at examples on Google. I'd suggest if first time you're using commands, do use the man command name, and that gives you help. And sometimes it does give you nice examples as well. So that's a good place to always do that. Always when you're working with data, always have a quick look at the files with the less overhead to double check the format and be sure that they actually match the input that you want it to do. And some programs are a bit finicky because they do require certain headers to be formatted in a certain way or a certain number of characters. It's always useful to look at your files as well and to make sure that all the data is there and intact. Watch out for data and headers, and you don't want to accidentally grab some if you don't want them. And a lot of headers tend to have special characters, so do be careful about that. And also look at your grep syntax as well. If you did something and you want to remember how you did it, a useful command to type is history, because that will show you then a history of all the commands you've used for the session, which you probably do today in today's practical as well. And do Google, do look out there, because I'm still learning new ways of using less and, more, and I've been using them for like 10, 15 years, just because different people use them in different ways, in different scenarios, and you kind of don't realize that you're going to use it unless you want to solve a problem. So it's always useful to look at message boards and how other people use it as well. So I'd just like to say thank you for your patience, and I'm sorry it's been a bit of a long talk, but hopefully it make more sense when you do the practical today. And I'd just like to acknowledge Dr. Amal Gulia, whose a lot of this training material has been adapted from, and the HE Banet IBT and NGS Bioinformatics course, Africa 21. Thank you. And I'm happy to take any questions.
Thank you, Sumir. We have time for some questions. So if you have a question, please raise your hand or put a question on the chat or also on the HackMD document.
Maybe also people I think have questions. Maybe I can ask a question. How many of the people here are first-time Unix users? If you can raise your hand. Okay, how many people have used Unix, Linux before? Maybe the ones who've raised their hands can lower their hands, and people who've used Unix, Linux before, if you could raise your hands. Okay, so we have a pretty good mix of people who have some Unix and Linux experience and some people who don't. So I think for the people who've got a bit of Unix, Linux experience, the practical will be useful for you, but it's introductory. So okay, that's pretty good, pretty good. So when it comes to using Unix, Linux, especially because the main way you'll be working with it is using a shell, so you'll have to do quite a bit of typing. So some useful things I would say is just be careful about like file names and stuff, especially when you're running applications, because a lot of the input-output errors you get is because it can't find the file. So your tab completion is quite important because that helps there as well as navigating to directories. Sometimes it might seem like an alien environment when you first start out, but it's pretty straightforward and simple when you start typing the commands and start doing what you want them to do.
Maybe I can ask a question. So
Sure.
maybe someone in the among the participants could be thinking that I'm only interested in just using bioinformatics for my data analytics. I'm not interested in becoming a bioinformatician. So why should I invest time in learning this complex dark screen? What would your response be?
Well, my response would be, okay, if you're going to do that, whereabouts are you actually going to work analyzing your data? If you're going to work in an institution that has a cluster on HPC, they're going to be using Unix, Linux as well. Do you need, I think if you're going to work with bioinformatics, especially with file and file formats, you do need to use Unix, Linux. Why? Because a lot of the file processing you'll do will be at the level of a cluster or an HPC. So you would need to do that. If you're going to do mainly data analysis, I mean, I think there are good packages out there like R. I think R or Python would be useful to use if you're doing mainly data analysis. But don't forget, in order to do data analysis, you need to pre-process your data because you can't just do data analysis on data that's not being pre-processed. And a lot of the Unix, Linux tools are very useful for pre-processing the data and also looking at it. And you can do that in R as well. I think if you're going to invest time in something and learn it well. Unix is nice because it's across all systems. And if you do work in any place that does have a bioinformatics capacity, you probably will, 99% of the chance you'll be accessing a cluster that has Unix, Linux on it.
When a cluster doesn't have a GUI.
Yeah, yeah.
Raised hands, do you have questions or is that your raised hands from the previous question? Jeremy, Audrey?
Sir, it was from the previous question.
Okay, that's perfect. Thank you. So if there are no more questions, I think you'll have more questions when you start working on the practical aspect of this. So look out for that. So we have a session coming up in the afternoon, introducing the HPC, which is what you'll be using for the most part of this training, and also during the other mini projects that you'll be undertaking after this. So having said that, I just want to appreciate Sumir for really the great introduction to Unix that he's done for us today, and he'll still be with us to take us through the advanced aspect of more or less the intermediate word said, grep and awk. So, okay, there's a question here. So, there's a question. Does that mean that tools like FASTQ alignment tools have not GUI?
Not usually, no. They don't usually have a GUI, but also one thing you have to remember with the GUI, if you're going to work with 10 or 15 files, it's fine to use a GUI. But if you're going to work with a million files, I don't think you can actually use a GUI. Because by the time you actually load all your million files in the GUI, your machine will probably run out of memory or any stuff like that. So some of the tools do have GUIs, but usually if you're working in this kind of scenario, you're working on a lot of data and you want to process all that data at one go. So using stuff like GUIs are not efficient at all.
There's another question. Can all genomic data analysis be done within the Unix platform?
Absolutely. That's how we do our own genomic data. We do it on clusters and within the Unix platforms. I think it's a lot of it is processing and also a lot of your programs because, well, once you get your input data, you want to make sense out of it. So to make sense out of it, you got to process it. So you turn that data into information. And a lot of the tools that you use, like your GATK and other things, all bundled in there and all work on the Unix system. And we tend to use Unix systems also because we're multiple users in an institution and we run lots of jobs. Like, if you're going to align, you're going to align like, 3 billion bases. And because you've got multiple users, the nice thing is then you have what's known as a scheduling system as well. That means then it allows everybody in the institute to use resources fairly without one user hogging all the resources, as you'll kind of see today. But yes, we do all our genomic data analysis within the Unix platform. I mean, if we're going to do statistical analysis, then I tend to do mine in R because I prefer R as an environment. You can do other things like with machine learning. You can also do that within a Python environment as well. The nice thing about Unix is it connects to other languages like Python and Perl and other things as well if you need to run stuff there.
Thank you. Any other question? Wonderful. So thank you all for joining and for your patience. It's really been quite an intense morning, and I know you've learned quite a ton. So we'll take 1 hour 30 minutes lunch break, and we'll be back at 2 p.m. East Africa time, which should be 1 p.m. CAT. Yes, and with that, can we have a round of applause for Sumir?